Hi everyone, my name is Ian Lee, and I am a bioinformatician at the National Biomarker Centre with Cancer Research UK. Today, I would like to share with you our current research on spatial profiling of small cell lung cancer using lentiviral gene ontology vectors and multiplexed imaging. Small cell lung cancer is a highly vascularized, aggressive neuroendocrine cancer. It has a high cell plasticity capable of transforming from neuroendocrine to non-neuroendocrine cells. It can metastasize to different tissues and organs, largely due to the high amount of CTCs, short for circulating tumor cells, released from this tumor, which promotes widespread metastasis and reduced patient prognosis. These circulating tumor cells can be collected from patient blood samples and can be used to generate CTC-derived explant models, and we call them CDXs, in mice. Although CDX were developed in mice, they retain the morphology and characteristic in patients, including the neuroendocrine and non-neuroendocrine plasticity in small cell lung cancer. To study the growth of the tumor, we also have an in-house resection protocol, which generates a paired subcutaneous and metastatic tumors for future analysis. One feature of interest of small cell lung cancers, which was also identified in CDXs, is vasculogenic mimicry. VM is a form of epithelial and endothelial cellular plasticity in which tumor cells develop vessels-like features, which allows them to transport nutrients and release CTCs into the bloodstream. And we currently hypothesize that metastatic cells may derive from a clonal population enriched with non-neuroendocrine cells and VM vessels. VM can be visualized by a combination of biotinylated tomato-lectin and CD31, an endothelial cell marker under immunofluorescence microscopy. At the same time, we can also use lentiviral gene ontology vectors, short name LEGO vectors, to track the clonal development of the tumor using immunofluorescence. LEGO vectors carries the three RGB colors, red, green, and blue fluorescence, and by simultaneously transduce them into CDXs, it causes the cell to express the fluorescence proteins at a random and unique level, giving each cell a unique color. These clones can retain their colors throughout the in vivo growth, and the colors of the different clones can be captured and analyzed using immunofluorescence staining. Now, although the color of the tumor is quite visible under the microscope, there is a computational challenge in conveying this visual information into a quantitative data for analysis. For example, the color intensity of the tumors may be affected not just by the clonality, but also the staining of fluorescence and the measurements from the microscope. Also, as the colors were introduced at a random level, it is quite difficult to accurately infer the number of unique clones from the colors, as the resulting colors exist in a spectrum of colors. Now, to address some of these issues, we have developed a color encoding pipeline for image analysis. After screening for the highest value colors, the color is then smoothed based on the adjacent cells. This encouraged the adjacent cells to have similar colors for clustering. Colors can then be clustered using k-means, and additional rounding towards the nearest tertiary colors may help remove some of the variations from shading or other variants. At the same time, there are various quantitative metrics for measuring the color diversity of the population, such as Gini coefficient and Shannon index. Gini coefficient is a measurement of inequality, and using this demo distribution, we can see that when the population is more evenly distributed, Gini coefficient tends towards 0. Shannon diversity index is a measurement of, diversity, and with this mock distribution, we can see that when there are some clones dominating in the population, Shannon index would reduce. So using these two metrics, we can describe the color, hence the clonal diversity of the small cell lung cancers. So using immunofluorescence staining and the diversity metrics, we conduct a pilot study to establish the growth kinetics and the analytical methods for small cell lung cancer tumors transduced with LEGO vectors. CDX17P tumors were grown in vivo and were harvested at different tumor volumes, including 2 small tumors and 2 large tumors. Clonal growth was observed in all of the tumors. We also noticed that there are no dominant clone emerging during the subcutaneous tumor growth, regardless of the harvesting size, and all of the tumors shows a highly diverse clonal populations. We then looked into the differences in clonal diversity of subcutaneous and metastatic tumors. Paired tumors were generated using our resection protocol along with Lego vectors. And although our previous single-cell RNA-seq results shows that the transcriptomic profile of the subcutaneous and metastatic tumor are very similar, we noticed that the metastatic tumor are only developed from a selected few clones, while the subcutaneous tumor is highly diverse in its clonality. We also looked into VM staining along with Lego vectors, and VM vessels were found to be localized to a specific clonal region, supporting our hypothesis that VM may be derived from a specific clonal population. And our ongoing studies will continue to focus on identifying these VM clones and their transcriptomic profile. So in summary, we have successfully implemented the Lego vector system in CDXs to track the spatial clonal development, and our pilot study shows a very different clonal profile and clonal diversity between subcutaneous and metastatic tumor. And to that, I would like to thank our team at the National Biomarker Center and our collaborators, as well as patients and their families. Thank you for your time in listening.